This method may be helpful in development of functional cosmetics and skin agents with anti-melanogenic activities, as they are easy to perform and the result can be obtained quickly.
In addition, this method may be useful for researchers who want to identify or investigate the effects of novel compounds or extracts with anti-melanogenic or pro-melanogenic activities.
To measure the tyrosinase activity, begin by seeding B16F10 melanocytes at a 5 × 10⁴ cells per well of a 24-well plate concentration in complete medium for 24 hours in a cell culture incubator. The next day, replace the supernatant in each well with DMEM supplemented with freshly prepared test compound, an inhibitor control, or a negative control, and return the plate to the incubator for another 72 hours. At the end of the treatment, rinse the wells 2 times with 300 microliters of cold PBS per well, and place the plate on ice. Next, add 300 microliters of lysis buffer to each well, using a cell scraper to collect the cell lysates after 5 minutes. Transfer the resulting cell particle suspensions to individual 1.5 milliliter microcentrifuge tubes and homogenize the lysates 3 times for 2 seconds at 14,500 RPM on ice to release the intracellular tyrosinase. Pellet the cell debris by centrifugation and transfer the supernatants to individual 1.5 milliliter microcentrifuge tubes on ice. Aliquot 70 microliters of each supernatant to individual